see.
Thank you very much. Very good morning, good afternoon, ladies and gentlemen. It's always challenging, isn't it, to be presenting and to have the whole lockdown, therefore forbidding you to go for lunch until the session is over. So I'm between you and lunch. Bear with me, I'll try to make it sweet and short. So today, I'll be talking about one particular strain, a Lactobacillus plantarum DR7. This is a part of our aging agenda. So our university is involved with aging development research projects, and we're one of the 3 universities in the country to do so. So before we start, let's look at some of the aging scenario. Now, we all know that the world is aging. This is a very old, I would say, statistics where we could see the darker the color, the more prone of the nation to go for aging. But what would that actually indicate? Now, this is Malaysia, where I'm from. If we look at 1950, that's how our population pyramid looks like. And as we go up the years, we see that the shape is changing, and it looks a bit more less triangle right now. Now, that is Malaysia, and this is Asia. A similar situation that we are looking at. And then now that we are in Europe, so how would Europe look like? Now, in general, most of the time we'll see that it's looking similar. But in actual fact, not so much. Now, this is my perspective. You may not agree with me. So basically, when we look at developing countries, I see it looking like the Burj Arab Hotel. If we are looking at developed nations, we are looking at like the control tower of the airports. Now, what would that indicate again? If we look at these different shapes, ultimately, it also shows a very different profile. Now, for developing nations, we can see population number growing. For developed nations, we can see it growing and that dropping again. So if we look at countries like Europe, Japan, Korea, population is actually dropping. Now, that is bad because we will have an increasing aging population, and amid this increasing Asian population, we have less people. What would that indicate again? Now, I see many young faces among us today. So the young ones, please pay attention. By the time you're old, no one's going to take care of you. I hope when I'm old, someone is going to take care of me. So the caregiver ratio is dropping. So ultimately, we are looking at aging and healthy aging. One of the alternatives is to look at probiotics, now that we are looking at gut modulation, immune modulation. Hopefully, probiotic can be one of the dietary intervention that could be used to elevate aging morbidities. Now, when we started doing this aging, we didn't know where to start. So we were planning how to do a screening, how to do a high throughput screening. Why not look at telomere? So this is a cartoon, as we can see. As we are aging, we're losing hair, so am I. So we're losing our crown of glory. And telomere is the same thing, it's going to get shorter and shorter. So do we have a solution for that? Well, we screen many. We screen more than 100 strains. And this particular one, plantarum DR7, actually showed a prevention of telomere shortening in aging rats. So we have the young rats, Y. Then we have the old rats, O. Then we have O plus statin, which are the old rats that were given this is our positive control with statin. And then also have aging rats and were fed DR7. So from there, we started. But what should we start? So as we can see, when we are aging, it actually affects many parts of the body, from head to toe. But we would like to emphasize on the brain. So first, we started with a very small model. And this model, many people are using it as a high-throughput screening again. So this is a fruit fly. They are very small. They're just the size of a tip of a pen or a pencil. What we did was that we have obtained a mutant, an Alzheimer mutant. So this whole fruit fly has amyloid beta expressed in its whole body. But they are flies. They can't talk, and we can't ask them how they are feeling today or have they lost their memory. So we have a promoter, a GAL promoter, that actually exemplifies the amyloid beta in the eyes. Now, this is a very nice photo of eye, positive control, and eyes of those what we call negative control with amyloid beta promoted in the eyes for visualization. This is the positive control. So in fruit flies' eyes, normally they look rather perfect, hexagonal ommatidia. They look round, but they're not round, they're hexagonal. And out of those hexagonal, they are actually bristles, and they're also pretty straight and organized. And this is our negative control. So in our negative control, we can actually see that the eye's morphology has already been defected. The nice hexagonal ommatidias are no longer nice. Actually, they've lost their shapes. The bristles, some of them are broken, some of them are malformed, and we could even see some holes or pores in the eyes. Now, this was the second generation of fruit flies that were fed with Lactobacillus plantarum DR7. So we saw some potential in that, and we evaluated further in its gut microbiota profile. And we found some quite interesting trend. Now, if we look at control and negative control, we could see a very drastic red color emerging. And that red color seems to be Wolbachia. So when we look at Wolbachia, we also have Y. Y is a commercial control, a very popular probiotic in Malaysia. And DR7, that's our strain. And we can see that the red color Wolbachia is actually being reduced. Now here we could see that the blue, which is the positive control, actually shifted more to the right, whereas the negative control shifted more to the left. Both the commercial control and also DR7 were in between, shifting gut microbiota profiles. And this is the abundance of different microbiota taxa. So, of course, we can see differences in positive and negative control here. However, when we were fed all with the different probiotics, we could also see a shift. And as a matter of fact, we could see more difference in bacterial taxa differences. When we did functional profiling using KEGG, we could actually see Wolbachia highlighted in green, having a myriad of different effects on different metabolic pathways. I am not highlighting everyone, but we're highlighting just 3 of it. And 3 of these are actually associated with brains, Parkinson, Huntington's, and Alzheimer's. We then proceeded to animal studies. So this is what we call an open field test. Those who are involved with brain studies and animal studies will be very familiar. So if the animal is having anxiety or stress, it would actually tend to go to the side of the boxes and hide or stop moving or reduce motility. But if they are active and so on, they'll be in the middle of the box. So when we look at the number of entries to the outer zone, and we could see that the all rats that were fed with DR7 showed a reduction in the number of entries to the outer zone. And as we know, as we are aging, we're also losing on neuronal branching. And that actually affects a lot of brain parameters, such as cognition and memory. So we wanted to evaluate that as well. This is a nice image of aging and young, as we can see, how abundance of neuronal branching. So we did we did several actually, but this one was more prominent in showing our data. We did what we call the Morris water maze. So our platform was hidden underneath water in a swimming pool. And the animals were trained for 4 days, and they were evaluated on the 5th day to see if they remember the way to find the platform. And I have a video. So these are the young rats, and after training for 4 days, we time how many seconds they used to find the platform. And this was one of the old rats, obviously forgotten. And then we have our positive control, O and statin. And finally, O and DR7. Excuse me. So when we look at the time that they need to find the platform, we could see that O was actually significantly higher. And DR7 and statin also reduced the time. Now, I always tell this to my students. This is a very important human analog. The swimming pool is like a big shopping mall. The platform is like me in a particular shop buying some clothes. And that poor rat running frantically, trying to look for the platform, but cannot find the platform, is like my husband. Then we also did another one for memory, which is called the T-test. So in the T-test, we have food at both arms, And these animals were also trained, and they have to remember which arm actually had foods, and that's the correct arm. So when we look at the time, how many percentage times that they entered the t-test and found the food, and we could see that for DR7, it increased the number of percent correct entries. Now, these are some of the inflammation markers. Do remember IFN gamma. So we have looked at some of the inflammatory markers, such as IL-1 beta, IFN gamma, TNF alpha, and we could actually see that statin, in some instances with our probiotics, could actually see a reduction effect in brain hippocampal inflammatory parameters. We also look at some apoptotic parameters to look at the possibility of affecting neuronal branching and hippocampal DNA fragmentation, and we could see that some of them that are involved in apoptosis were actually reduced by Lactobacillus plantarum DR7. We also look at senescence. This is for senescence. So P19 and P53 is one of the biomarker for senescence. Since we were dealing with aging animals, so we think that we should opt to look at this. So P19 actually inhibits P53. P53 actually have an effect on apoptosis and senescence. Now, when we look at our data, we found that P53 was actually downregulated by DR7 and statin, the positive control, and P19 was actually upregulated. And we postulate that's because P19 need to suppress P53. We also look at brain neurotransmitters. Now, we are aware that brain science is very complex. There are many mechanisms and there are many possible outcomes on why this happened. But let's target some of the brain neurotransmitters. Now, in brain, we have 4 main neurotransmitters that goes through GABA, dopamine, norepinephrine, or what they are called sometimes noradrenaline, and the last one, serotonin. So we tested one by one. These are gene expressions of the hippocampus. We didn't see anything significant in terms of our strain for GABA, TH, and DBH. TH is the enzyme that converts tyrosine to dopamine, and DBH is the enzyme that converts dopamine to norepinephrine or noradrenaline. So we didn't actually see any effect. However, when it comes to the serotonin pathway, we did see something in the animal studies. So in brain, tryptophan can be converted to kynurenine or serotonin. So under stress, in many instances, it has been found that tryptophan was converted to kynurenine via the actions of IDO. Now, on the other hand, if tryptophan is converted to serotonin, it would be going through the actions of TPH. So when we are looking at the gene expressions of both these enzymes, we found that the gene expression for IDO was actually downregulated as compared to TPH, which was higher than the old negative control. So we believe that tryptophan along the serotonin pathway may be one of the biomarker or pathway that was affected by DR7. So we then proceeded to a human study, which was published this year. Yes, published this year. Now, instead of directly going through human studies for the aging population, we wanted to see if there's a possibility that we can look at a younger population, and hopefully that we can look at probiotics as a dietary supplement to actually have a healthy aging process right from the young towards aging. So what happens? This is a double-blind, placebo-controlled, randomized, parallel. What we did was that we screened all our subjects for moderate stress. We used the PSS-10 questionnaire. So if they are not stressed, then obviously they don't qualify. And believe it or not, there are so many academicians that don't qualify, not stressed. We have placebo 55 and DR7 56. So it was 12 weeks, and consumption of DR7 was for 9 log CFU per day. So when we look at all the stress parameters, we had PSS-10. We also used the DAS-42 questionnaire for stress, anxiety, depression. However, we didn't see significance for everything. We only see it for 2. Stress and also anxiety, and these are both from the DSS-42 questionnaire. And when we separated the age group, we also found a very not very different, but a different trend that we see from an overall subject. So we actually separated the subjects by age, and let's see what happens. So we separate them to below 30 years old and above 30 years old. And what we have found was that DR7 actually affected those young adults, what we call below 30 years old, more in terms of stress. And then they affected the older adults, not so old, above 30, in a more manner of anxiety. And we were thinking, this may be because stress, there are many stresses. Being stuck in a traffic jam can be stressing. Sitting here can be stressing. As you're hungry right now, we have the door locked down for lunch. So there are many stresses. Now, if we do not manage stress well, stress will eventually progress to anxiety. And again, if we do not address or manage anxiety well, then anxiety may progress to depression. But in our case, there was no effect for depression, only stress and anxiety. We postulate that the younger people have higher stress levels, and that was maybe why DR7 targeted that. And as they grow a little bit older, maybe their stress management may not be that well, and it progressed to anxiety, which was why at a little bit older adult, we saw the effect in anxiety. So this is how the DAS42 questionnaire looks like. It has 42 questions, and 14 questions for stress, 14 questions for anxiety, another 14 questions for depression. So this is for stress. Now, when we compare week 0 and week 12, how much stress scores were different? So if stress scores dropped, then it would be indicated by a whiter color. So as we can see from DR7 and placebo, we could see a roughly more whiter color. Now this is just an illustration. The p-value was indicated in the table just now. Now that was for stress, and this is for anxiety, similar nature. A whiter color indicates a more dropped in levels, anxiety scores. So we see a more prominent one in DR7 and placebo. Then of course we did cognition. We did a lot of parameters for cognition. I'm only showing half of them. Now, we did not see a lot of significance for a lot of these parameters. We only see some very minor cognitive improvement. One example would be the social-emotional task and also associated learning. So we saw a little bit of the improvement here, especially in the older adults that were above 30 years old. Then we also look at some of the inflammatory markers again. So the top one would be for those younger adults, age less than 30, who are experiencing stress. And we could see that cortisol, stress hormone, was actually improved. IFN gamma, IL-10 was also improved. Those older adults, age above 30, we could only see 2, IFN, TNF, and IL-1 beta, primarily all the pro-inflammatory biomarkers. And in all subjects, we see quite a few of them. Now, we were talking so much about neurotransmitters in animal studies. We want to see if our animal data could actually be translated to human data. So we also took whole blood and did gene expression. And what we saw was that for those less than 30, those above 30, and overall subject, 2 very prominent parameters were actually upregulated and also downregulated. So if we look at TPH and IDO, there were 2 of them. So TPH was actually upregulated. This is an inverse data. And IDO was actually downregulated. So what would that mean again? So we made a summary of this. So for placebo, in terms of the serotonin pathway, we actually saw an increase in IDO or TDO, but a decrease in TPH, indicating a higher reduction of tryptophan to kynurenine and less of tryptophan to serotonin. However, in the probiotic group, the DR7, we see an inverse effect. In dopamine, we didn't see much. In the DR7 group, we postulate that dopamine was actually maintained. So these were 2 of the pathways that were targeted. So when we look at the gut-brain axis, I think we have seen several slides already talking about all these connections. So again, we like to stress, yes, indeed, the gut-brain axis, the BBB, they are very complex. And there are so many metabolites that we're looking at, so many different pathways. So in terms of our studies, what's possible? So remember, we are talking about IFN gamma just now. So in terms of animal studies, in terms of human studies, we saw this as a very prominent biomarker that was affected. So in terms of stress, in terms of gut-brain axis, we are looking at IFN gamma that is actually affecting IDO. And IDO, in turn, in the brain, actually affects the conversion of tryptophan to kynurenine. So during characterization of our strain, we actually found two compounds. Let's just call them compound A and compound B. Now what we did was that we did a preliminary docking. So this is ongoing, and what we want to try is to understand what actually happened. Now when we docked IFN to IFN gamma, which is the IFN to its own receptor, we see an energy level of negative 16,000 plus. So basically, if two compounds can bind together better, it indicates they need less energy. If they need a lot of energy to bind, it would indicate that that connection is not so strong. IFN 39 is a known inhibitor of IFN gamma to its receptor. The controversy here is that we saw a very much reduction in binding energy, indicating that once IFN 39 binds to the receptor of IFN gamma actually binds better. So how is that possible? We look at two of the compounds that we have isolated, compound A and compound B. They actually increase the energy of this docking. So what does that show? This is our cartoon. So this is IFN docking to IFN gamma docking to its own receptor. It yielded an energy of negative 16,000. Then, when we have IFN 39 is a known inhibitor. It's this color. So what happens is that once it's bound to the IFN gamma receptor, IFN gamma can still bind. However, do notice that the orientation changed. And look at the binding site. Binding site is here. The binding site has already changed. So yes, no wonder binding energy was reduced. It could still bind, but at a different location. And therefore, IFN gamma was not activated. And that is also why IFN 39 was a known inhibitor. So for both of the compound A and compound B, had a similar effect. First, the orientation was changed. Second, energy was increased. And third, binding site was also affected. So we believe these two compounds may have potentials. And we would like to look further into this later. Actually, I just got this data 2 weeks ago. Okay, so Lactobacillus plantarum DR7, patent for this has already been filed in Malaysia, but patent is ongoing in Korea and also in China. Patent, of course, the culture was deposited for patent depository. Whole genome sequence for DR7 is on NCBI right now. Human study on stress, memory, cognition, that's the one that I showed, published. URTI also published. Gut not published yet, we just received the data two months ago, but yet to be analyzed for pyrosequencing. So the reason why I'm showing this slide is because we do have quite strong connections for collaborations in Asia. I wish to establish some connections in Europe. So if any of you are interested, please contact me. I'll be happy to join. Now this is the main conclusion. That's the end of my slide. So it's okay to age, but do age healthily. So ladies and gentlemen, thank you very much.